Okay, so thanks, Mikhail, for the introduction. I'm Jaime, and I'm working as a postdoc here in Norway, and most of my research has been around sequence motifs. I mean, I have been working on developing methods to study transcription factor binding motifs, and so I feel comfortable talking about this. And that's the reason that I accept to give this seminar. So I will not present exactly what I'm doing. I mean, I'm not presenting my research, but instead it's just a summary of the methods. And the message I want to transmit to you is give you an overview of this field. What are the methods that we can use, and what are the questions that we want to address in the future, or where the field is moving. So it's just a general presentation. So feel free to interrupt at any time. So don't be shy and just unmute yourself and ask questions. Okay, so just a little thing before. Most of the examples here are about transcription factors, because that's the field where I'm working with, but many of them can be applied also to RNA sequences or protein sequences. So, but I mean, this is just general, and the same methods can be applied. Okay, so let's begin with this. So the basic question here is, what is a motif? So in genomics, it's a pattern found in a set of biological sequences. And I highlight here genomics because in biology, we have patterns occurring in other parts. For example, if we talk about networks, then we can find new network patterns, or patterns of development or segmenting, for example, in flies or in other organisms. So here, so even this can be patterns from Norway or from Mexico. So here, this column here represent are motifs, but they are represent on different ways. And we will go going from simple to more complex representations. So the very simple one, and don't recommend it at all to use it, is string matching. So maybe you will find it in some papers, and it's the simplest way to represent a string in a text. So it's when you, for example, type Ctrl+F in a text editor, and then you type a word. This is what this is the kind of matching that you are doing, and it's very simple to use. But the problem is that here, so there are two problems, I would say. So the first one is that we have to type word by word, so one by one. And the second and more problematic is that we are not comparing against a background, against a reference. So I just mention it because I have seen this in publications, but never use it. I mean, it's just to know that it exists, but not recommended at all. So when we have, another way to represent sequences, I mean, also for proteins or RNA, is that we can use a consensus. In the case of the DNA, we can use different and a specific alphabet to represent the different combinations of nucleotides. So the motif is a model that it's representing a set of sequences that have something in common. So, for example, that they are recognized by the same protein, or for example, the transcription start sites or splice sites. So, and we want to have one model that represents these sequences that have something in common. So a simple and informative way to do this is using the consensus, where we can we can use this alphabet. So for example, if we consider this column, we have a C or a T. So this is a pyrimidine, and we can represent it with Y here. Or another way to see the consensus is like a regular expression of a biological sequence. I mean, like the regular expression that you can write on any computer language. So the advantage of the consensus is that we can represent variability and ambiguity in a string, and many sequences can be represented as a single expression, as in this example. But again, at the end of the day, this is a grep search for many words, and the problem is still the same. We do not have a background to compare a contrast. So, but we will see. So we will see later how to solve this problem. But consensus are always very frequent in literature, and my advice is that you can use it for sequence representation only, but not really for detecting new binding sites. So if you find something like this, it's okay, just for representation. And this you will find it in many papers. And here are some examples. So here is the binding sites of the consensus for FOXA1 and ESR1, two famous transcription factors in breast cancer. So this is the UPAC consensus, and this is how will be represented using a regular expression. And here we have the logo that we will see later. Where does it come from and how to interpret them? So, okay. But before moving to more complex models, I want to highlight something that we always are, it's not exactly a problem, but it's sometimes things that we have to do before analyzing motifs. It's calculate the sequence complexity. In this context, the complexity is related to the possible number of words that we can obtain when we shuffle a sequence. And these low complexity sequences are, for example, short repetitive elements, for example, microsatellites or sequencing artifacts, maybe for contamination. And many of them have a biological function. But the problem is that sometimes they make the analysis difficult. So for example, when we want to align sequences or assembly genomes, and they can be confound the sequence analysis. So before trying to discover motifs, one way is try to identify or try to, like a sanity test, for example, to say if this sequence is complex or not. And as I told you, the complexity is related to how many words can I generate, how many new words can I generate if I randomly shuffle this sequence. And to calculate this score, there is one formula or one score. It's this one here. I forget the name. It's very difficult to pronounce. The Wooton-Federhen complexity score. It's basically calculating the number of permutations. And even as I mentioned here, that many of these low complexity sequences may have biological function. This is one example. So this transcription factor that it's called, or transcription factor from the same family, the Kroppel-like, they tend to have to bound low complexity sequences. And here you have some examples. For example, this sequence TA, or GGG, GAGAGA, all of these kind of sequences are of low complexity. Here is one example, or many examples that I calculate using that formula. So, for example, a poly A, if we change the position of we randomly shuffle the A's, at the end we obtain the same word, so the complexity is 0. And of course, here we have the complexity for other transcription factor binding sites. So for example, for CLEF, it's this one. For CTCF, one of the largest, one of the transcription factor that binds largest binding sites is this one, and you see that the complexity is higher. Even if when there are many positions that are similar, but this is a way to have an estimate of how complex. And just to mention this, the score from this formula goes from 0 to 1. So it can be in a way to compare sequences of different sizes. Okay, and now we go to the third model, that are the position frequency matrices, or position weight matrices. That you can find them in the two ways, but they are different, and I will show you why. So I think this is the most used model to represent biological sequences. And we can say that this is a probabilistic representation of the sequences. And it's a simple matrix representation that can be used for many, not only for transcription factors, but for example, can be used for representing RNA or amino acid domains, splice sites, transcription start sites, or many other elements, sequence elements. So I think it's very intuitive and it's a simple representation. And in contrast with the consensus or the grep matching, this model also integrates the background, and I will explain you in a few slides. Okay, but of course this is a model. This is far from being perfect, and of course every model is not. They have some details, and the problem here in this representation is that we assume independency on every position, on every position in the logo here. So the problem is that at least in nucleotides, we know that there are there are interdependencies of contiguous nucleotides. So the classical example in vertebrate genomes is the CPGs, and these dependencies we are not consider them on when we use this model. So this is one of the big disadvantages, and it's not necessarily applied for all the transcription factors, because in many of them this assumption of independency is correct, but in others we observe dependencies, and in these cases we need another model to really to have better predictions for binding sites. Okay, so now a few slides how we obtain these matrices. So first we need a collection of sites. This could be sites detected computationally or experimentally, and the condition is that they have to have the same length. So in this case we have 8 sites of size 14. So the first thing, as I showed you before, we can create a consensus sequences from this alignment of binding sites. But I told you this is only for representation purposes. And then we can count how many nucleotides we have on each position of the alignment. And then and this representation like this is what we call the position frequency matrices. And this is the model that the most common model, and what you usually find on databases. Yeah, they're here, such as Jaspar, Hokomoko, SysDB, RegulonDB. So this is the most used model to represent sequences. It's very simple, and you see that it's very intuitive. So just count how many nucleotides at each position, and that's it. However, in many papers, maybe you have seen them, they use indistinctly position frequency matrix and position weight matrices, which is not exactly the same, because actually the weight matrices comes from the frequency matrix, but we need something, we need a reference to really calculate the weight, and this reference is the background model that I mentioned before. So just to make it clear, the frequency is just count round numbers, and then the weights is when we use the background, when we integrate the background information. So, but what is the background? So the background is just probabilities, or the expected frequencies of nucleotides or amino acids, and this will indicate us what are the sequences that have higher chance to appear just by chance. Of course, these values may change from sequence context. So, for example, if we calculate well the nucleotides, if we count the nucleotides on exons and on promoters, for example, or in I don't know, in repetitive sequences, they will have different probabilities. And of course, we can have higher or we can use Markov change in order to model these backgrounds. So for example, we can calculate probabilities of D nucleotides, 3 nucleotides, and higher order. So these are two examples, very simple. So what are the probability of observe an A if we take the whole human genome? I mean, the mononucleotide model. But also here we have the probabilities of observing D nucleotides in human promoters. So you see that they are very different, and I will show you one example that you see that why it's important to choose correctly the background. So this is one of the most tricky parts on the field, that it's very important how you choose the model. And here, for example, you see that this is a C followed by a G, so it's a CpG. So you can see that the probability of observe a CG in human promoters is lower than other dinucleotides. And this is something known in biology, that promoters are depleted of CGs. So if you use a background with higher Markov order, you will be closer to reality. And the same thing, and if you model with a very simple background using mononucleotide, you lose this information. So as an advice, if you are working with DNA, you should use at least D-nucleotides as background, because this already includes these nucleotide dependencies. Just that it's not always possible to obtain higher order background models, but because of course we need more sequences, and sometimes it's not possible. So for example, if we are working with bacterial genomes, maybe the nucleotides it's possible, but maybe not trinucleotide or tetranucleotides. So this we have to adapt it depending the organism that we are working with. But as a rule of thumb, just try to use at least the nucleotides if possible for the background. Okay, so here's one example. So we are calculating the probability of observe this sequence using the two backgrounds. So here we have probability of AA, so A2 times, squared, probability of T, because we have the T2 times, and C and G, and we have this number. And then if we use the probability, if we calculate the probability of observe this sequence using the dinucleotide background, so we take the A here, and then what's the probability of observe an A followed by a T? So it's this one, this number goes here, and then a T followed by an A, so it's this number here, and then you can continue until the end of the sequence, and then we have these two numbers here, and you see that there is 1 degree of magnitude of difference, just because we are evaluating the same sequence but using two different background models. And this is very important, and it's something that you should have always in mind when you are working with Motifs. The background is maybe the key part of the analysis to choose correctly. So okay, so coming back to the position frequency matrices. So when we calculate the weight, it's just this log ratio between the frequencies or the probability of observe a word given the probabilities here, divided by the probabilities that we calculated using the background. And this is what we call the weight. And then we can take, now we have the weight, we can take any sequence of the same size, in this case of size 14, and then we can assign a score of this to this sequence. So for example, if we take the sequence here and the weights here, we will obtain a score of 5.23, for example. And this is another way to represent the same formula. So it's a log of two probabilities. The probability of this matrix. I mean, to convert this matrix into probabilities, we have to divide it by the number of sides. So in this case, it's 8. So this will be 1. This will be 0.5, 0.5, and so on. And then and divided by what we calculated on the background. And that's the score of a sequence. And then we can try all the sequences, or we can scan all the sequences in the genome or in a set of for example, ChIP-seq peaks or promoters or any kind of sequence that you have. And then we can slide this matrix and detect what are the positions that contain a weight above 0. So that means if the weight is above 0, that means that this sequence has a higher chance for appear using the matrix than the background. And then maybe this sequence is a potential binding site. Of course, it's sometimes what makes sense in statistics maybe makes no sense in biology, right? But this is just a model, and then of course there are many things to try to really be sure that this is a true binding site. But this is only math. And okay, so the point is that with this weight we can score any matrix, any sequence of the same size, and then the highest the score is the most similar this sequence is to the model. And okay, and we want to use this matrix to detect new binding sites that are very similar to, for example, here we start with 8, but maybe 8 non-binding sites, and then we have a sequence that we don't know, maybe a new genome, and then we want to detect if this genome has binding sites for this TF. But okay, so the information content, or it's a way to, the Shannon entropy is a way to measure uncertainty in a model. And it's a very simple way to calculate it, and this formula maybe you will see it in other in bioinformatics very frequently. So it's very simple. Don't feel intimidated. It's just the probability of one event times the log2 of that probability, and of course it's a summation of if we have many probabilities. So and we are measuring uncertainty. Right? So let's say that we have a 4-letter alphabet, so the 4 nucleotides. And let's say that each one has the same probability. So we have complete uncertainty. So we don't know nothing about the model. So if we do this, if we have no uncertainty, then we can calculate the maximum entropy. So in this case is one quarter, it's 0.25. So times log2 of that number, and we sum it 4 times because we have 4 nucleotides. So if we make this calculation, we obtain a maximum entropy of 2. If we have uncertainty for 2 letters, so it's distributed in 2 positions. Okay, if we have the probability distributed in 2 letters, so it's the maximum entropy is 1. And if we have all the probability distributed in only 1 position, that means that we have no uncertainty because we know that all the probability only goes to 1 nucleotide. The maximum entropy is 0. So then, using these special cases, we can calculate the information content. And this information content is a subtraction of the max entropy, so in this case 2, minus the uncertainty that we observe in a given position. So this is, can you change it, please? Yeah. So this is only for DNA, but now for nucleotides, for amino acids, now we have 20-letter alphabet. So if we do the same, if we plug these numbers on the formula, the max number is 4.32, and that's the reason that in protein logos, the y-axis is higher. Okay, can you move one? Yes. So now we have an example. So this is the same FOXI1 motif that I have been showing you during the presentation. And here on top, we have the position frequency matrix. So now let's calculate the information content for the third and the 12th column. So remember, the maximum entropy for nucleotide, for an alphabet of size 4 is 2. So it's always 2 plus minus, depending the sign, of these numbers. So then we plug these numbers in the formula. So it's 1 over 500, that is the total of sites, times the log 2 of that number, and then we sum it for the four rows here, and then we obtain 1.97. And that's why this third position, it's very close to the 2 bits of information. And in terms of uncertainty, we are saying that we are almost sure that this position will be a T. So there are a little of uncertainty. And by contrast, on the 12th column, we have a little of all the nucleotides. So when we plug these numbers on this formula, here you see that it's very low, and it's telling that this position, this 12th column here in the matrix, does not contain, is not informative, and this is reflected on the sequence logo. Next one, please. And you can see, and this is just some examples of logos, and where, for example, this one on the right are very, let's say, bad quality because they have no information content, or well, very little information content. And those on the left side are more informative, and then they are more trustable, let's say, in a way. Okay, next one. And so I will not present the details of the motif analysis algorithms, but I just share with you these links. So these are the at least motif discovery that I have used before, and many of them they run on different ways. Some have some advantage that others do not. So my advice is that you don't stick to only one. So maybe try different, because sometimes you will find motifs with one but not with the other, and vice versa. But all of these algorithms are working with the position frequency matrix. So that's in a way interchangeable. So for example, you can run something with Homer and send the results to programs in MIM, or from MIM to RSAT, or vice versa. So these are only for position frequency matrices. And in general, I would say there are two families of algorithms. So for example, Homer and RSAT, they have algorithms that count words. So for example, they create a list of all the possible k-mers of a given size, let's say k-mers of size 6, and then they count it, and then they compare the observed versus expected, given a background, of course. And the other, Lambda, Chipmunk, and MIM, are based on expectation maximization. So they are trying to adapt a mixture model on sequences. And from this, I think MIM is the most popular, but there are other nice alternatives that are working in a different way, and they are, I would say, as good as MIM. And these algorithms are not new. Actually, they have, I would say, 20, 25 years on the field, but they are still they are still relevant. And one reason that they are very used is because the model itself, it's a matrix, and then you can use this, and they can interchange the matrix from one algorithm to another. Okay, let's move to the next one. So now the next part is the complex representations of the motifs. So as I mentioned before, the position frequency matrices have this weak part that they are not modeling interdependencies on nucleotides or amino acids. So there are new models that came out more or less in the last year, in the last decade, and here they are trying to model dependencies. So here's one example based on hidden Markov models. And here my personal opinion of these models is that they are more difficult to interpret and not always are very easy to visualize. So for example, here, if we follow the 7th column in the top motif, we have an A, and the other positions are white. It's saying that we mainly observe an A on this position, but the position before, because here we are modeling dinucleotides, will be a C. And that's the reason that the C in the 6th column, it's always in the 4 rows in the top part. So these are called transcription factor flexible models, but this is only one way to represent. Can you move to the next one, please? And more recently, there is another extension that it's called the Bayesian Markov models. And here they make it general, so they are representing the mononucleotide logos, the position frequency matrices, in dimers or in dinucleotides or trinucleotides. And you see how the information content decreases. And this is just, and there you have a formula that it's saying if you, as more as you increase the Markov order, it's more difficult to see the actual information in the logo. However, the next slide, please. You can use the same method to visualize very long sequences and identify relevant patterns. So here they are modeling poly A tails, and this is the picture on top. This is the mononucleotide, the position frequency matrix, as we have seen before. But then you can apply this method to visualize the dinucleotides or trinucleotide dependencies. So I have tried this method, and for me, this is just an extension of the position frequency matrices, but instead of having 4 columns, now we have 16 because the 16 the possible dinucleotides, and the calculation is the same. So we can sum all the sites that we have in one column, let's say in the AA column, and divided by the total. And we proceed in the same way as I showed you before with the mononucleotides. But of course, now we have much more parameters than before. Next one. And yes, so, and of course, These are only a few examples, but as we know, this is a probabilistic model, and sometimes we tend to forget that DNA is actually a molecule with physical properties. However, there are ways to add this information when we want to detect transcription factor binding sites. And in this publication, and well, actually, first, there is a program called DNA Shape, where you can train a model that gives you different properties of the DNA, like minor groove width, or the twist on the DNA helix, and other properties. It's called DNA Shape. And you can use these features plus position frequency matrix to try to improve the transcription factor binding site prediction. So the curve here, the plot here on the right side, shows the precision recall curve. In x-axis is the predictions that we have using the position frequency matrix, and in the y-axis we have when we combine the matrix plus the DNA shape information. So the message here is that for many those points that, and each point here is a position frequency matrix. So the points are that are in the diagonal. So that means that if we add the DNA shape features, we are not seeing an improvement. But in other cases, the points that are not close to the diagonal, that means that if we add the DNA shape information, we have a better performance. So we have a better predictions. And so yes, adding DNA shape improves binding site predictions, but not for all transcription factors. And this is something that has to do with related to the biology of the TF, because some transcription factors indeed recognize not necessarily the sequence, but the shape of the DNA. And these are for some specific families. So the next one. Okay, before going to, can you go one slide? Yes, no, before. Yes, so these are only a few list of methods that I know, but or that I have used before. But of course, there are many methods more and specialized on different properties. So, for example, these are more general, but there are some specialized to work with particular transcription factor families and so on. So, but this is just an overview. But you can contact me if I can give you more a list of tools for and to use it for specific cases. Okay, one more. Okay, and the most recent ways to obtain sites and bind motifs is using deep learning models. So I don't know how familiar are you with deep learning, but to summarize it, is just one way to classify things to classify objects or images or sequences. In this case, the input are the sequences that you have on the right side, on the left side, and the output is that we want to send each input sequence. We're going to classify it in two categories. For example, if the input sequence bound by the transcription factor or unbound, for example. So the point here is that these kind of methods allows to know exactly what nucleotides on the input are pushing the prediction towards one class or the other. I mean, next one, please. So here we have on top we have 3 sequences of size 200, and we see sort of logos, but they are not exactly information content. The y-axis here on top is something we call a contribution score. If the contribution score is positive, that means that they are pushing this sequence to the, let's say, positive class. And if the contribution is negative, they are pushing the sequence to the other class. So the advantage of this kind of method is that we can focus on longer sequences. In this case, you can see that they have 200. And from these 200, we can see that what are the exact nucleotides that are contributing to classify this sequence in one class or the other. So we can trim these long sequences and at the end get the sequences that are on the red boxes on top. And then we can cluster the sequences and aggregate them to finally construct motifs that will look like the position frequency matrices. But they are not, I mean, they look the same, but it's not exactly the same because they are derived from importance or contribution scores. But other nice thing that you can do using this kind of models is that you can change what it's on top, on the bottom right. So you can change nucleotides on your sequences and reevaluate the mutated sequence and see what happened with the prediction. So for example, you have on bottom right, you have something that says C to T breaks Rela motif. So that means is that you change one of these letters by a T. Now this part of the sequence is not predictive anymore. But sometimes some mutations may make the sequence more predictive than before. So this is called in silico mutagenesis, and this is one thing that you can do with deep learning models and not with the classical ones. So this is one advantage that these methods have over the others. And these importance scores can also be not only from can be obtained not only from deep learning, but also from support vector machines or non-negative factorization models. Okay, so let's move to the next one.
Sorry, there's a question in the chat asking if this paper are bioarchive paper or has it published somewhere?
Yes, so actually there are two papers. Last week was released one of these new papers. I don't remember the algorithm. The algorithm is called BPNet, so you can check. It's now on. It was on bioarchive, but now it was published. And the other, it's called TF Modisco. I will write it in the chat. From the same authors. And those are the authors that developed these contribution scores. So, yeah, someone posted. Yes, so I think those are the state of the art right now on sequence prediction. And of course, this BPNet is for nucleotides, but you will see that there are many other papers coming right now using deep learning to try to identify sequences, not only nucleotides. So I would say that they have one very big advantage over classical models is that you can detect, since neural network is also detecting nonlinear relationship on sequences, you can detect other patterns such as grammar or what we call motif grammar. So like the orientation or combinations of binding sites on the same sequence. So this deep learning model allow to detect that the classical models do not. So this is a very big advantage. But of course, one disadvantage is that you need a lot of sequences to train it. And maybe you cannot apply this to all organisms because you don't always have a big amount of sequences to do that. But in general, they work very, very well. Okay, the next one. Okay, so just to conclude, of course, the modeling of sequences is an old field in bioinformatics, but it will be always relevant because at the end, that's what we are working with, only with sequences. The position frequency matrices are still the most used model even when I have shown you now that there are many other alternatives that are more complex and maybe more precise, but they are also becoming more popular. So these new representations are not as popular, but we already show that they improve binding site predictions, in particular for some families. But what I see that why they are not so popular is because they are not interchangeable. So, for example, as I mentioned before, if you run Homer, then maybe you can continue your analysis with other program developed by MIM, because the format is the same. It's just a simple matrix. But here, in one model, you represent the nucleotides, and another you represent contribution. So it's not interchangeable, and you require many more parameters to train one model and compared to the other. So there is no really interchangeable. That's why I think people still prefer the simple models. And of course, also for visualization things, because the matrix we can print it as a logo, but then we cannot print, let's say, oligomers or more complex or dependencies between nucleotides. But of course, the more complex the model, we can obtain other kind of relationship as the motif syntax. And this is also very important. So just to say that my final word is that there is a lot of room for improvement, because most of the methods so far have been designed for position frequency matrices. But now that the more complex models are becoming more popular, I think there will be, I can see that there will be many tools around these new models, and then that's where we can make a little difference on this field.
Okay, Jaime, so there's a question.
Yes.
Larger scales, for example, or over 100 base pair, is sequence specificity competing with other factors like molecular crowding, chemical properties, in determining DNA binding, and also can neural network incorporate those information?
I mean, I think yes. I mean, can you come back a few slides?
Okay.
On the DNA shape thing. Yes. Yeah. So at the end, the DNA thing, the DNA shape, it's also vectors of the same size as your motif, and on each position of the position frequency matrix, you will have a given value for each feature of your DNA shape. So now you can, if you move to the next slide. So now what you could do is add to your sequence, because at the end the sequence is converted in a numerical way for the neural network, and then you can just, I think you could concatenate the additional shape features, and maybe this will, and I think that's something very easy to do. I haven't tried, but I think that it's possible to do. Of course, the interpretation will be different, but I mean, I'm not the expert on deep learning things, but I think it's possible just simply adding, I mean, it's just one more, just make it a little bit, the input vector a little bit longer. Yes.
Okay. So there's another question. For RNA, is known to adopt a variety of secondary structures. Do you think these sequence-based motif finders are a good option to detect motif for RNA binding proteins as well? And do you know any of those methods?
There are methods that are specific to RNA, because let's say the the base complementarity in DNA is not the same as in RNA, so the constraints should be different. But one method that works well with DNA and RNA, it's called deep bind. Yes, deep bind, and this was the first method doing deep learning on genomics, and I think it's one of the cornerstone papers that after that paper, many people start to work with neural networks on genomics, and that method was able to work with RNA or protein. I mean, of course, maybe a slight difference maybe in the network, in the neural network, but was able to work with both cases. So yeah, it's possible.
Okay, if there's no question, I have one, probably a very simple question.
Mm-hmm.
So you mentioned about the background model that we can use to determine this position weight matrix. And when we are choosing such background, do we need to consider, for example, the regulatory region we are interested in? For example, if I'm interested in the binding and promoter region, then should I simply use all the sequence in all the known promoter, or should I maybe use the whole genome as my background model?
So normally so for example, let's say you did a RNA-seq experiment, and then you detect your differentially expressed genes. So what you can do is select, let's say and then you want to make a motif discovery on the promoters of the differentially expressed genes. So the background that I will use will be all the promoters from all the genes, because the idea is to try to keep sequences that are in the same context. So normally many people use this, like all the promoters when you are focusing on promoters, or for example, use all open regions on the genome, for example, from the DNAs or ataxic tracks. But using the whole genome as background is not good, because, for example, you will have many places that are very repetitive, and this will maybe have create a bias on your background. So, for example, especially if you are in the centromeres or telomeres that you have very repetitive, I think the background there is very different than the background on the promoters. But it's never recommended to use the whole genome as a background. So always use the most similar sequences, the universe of more similar sequences. So, for example, if you have 200 differentially expressed genes, the promoters of these 200 genes, then take the universe that will be all the promoters.